Hello children, today we are going to discuss about the resolving power of optical instruments, right? So what do you mean by resolving power? This is something new term for 12th standard. Let us understand what is the meaning of resolving power and how do we resolve the images, right? So we will discuss about resolving power first, then we will discuss about resolving power of compound microscope, and finally we will discuss about resolving power of astronomical telescope, right? So first of all we will understand that what is resolving power. Suppose there are two objects kept at some distance from an aperture. Let us say this is object 1, this is object 2, and there is an aperture, right? Now this aperture has size and there is a screen here. We have kept a screen here. Now the light coming out of this object will go till here. The light coming out of this object will go till here, right? And say this is the size of the aperture. So let us say this size of aperture is denoted by D, right? This is object 1, this is object 2, because we have learnt in diffraction that light bends around the corners of the obstacle. So if I am taking this as some obstacle whose aperture is this much, D is the distance. So the light coming from here will bend and reach till here. Light coming from here will bend and reach till here. So that is what aperture size. Beyond the aperture size, the light will enter in the geometrical shadow region of the apertures. So that is what we have learnt in diffraction. Now suppose if the light is reaching in the aperture region, let us consider this as the center and let us say this was the aperture. Distinctly we will draw it so that we can easily identify how this angle is made. So let us name this angle as delta theta, right? So due to this light there will be some diffraction pattern like this. Due to this light there will be some diffraction pattern like this. Now we are able to see the image formed by object only if they are distinctly made, right? And this ability of formation of two distinct images is called as resolving power. Say this is the diffraction pattern of one, this is the diffraction pattern of one. Iska image alag or iska image alag. If this would not have been the case, if the diffraction pattern would be like this and like this, तो एक का इमेज ऐसे है, एक का इमेज ऐसे है, ये दोनों हमें डिस्टिंक्टली नहीं दिखाई देंगे, राइट? द इनएबिलिटी ऑफ फॉर्मिंग द डिस्टिंक्ट इमेजेस, दो ये ऑब्जेक्ट्स हैं तो हमें वो अप्रोक्सीमेटली एक ही इमेज दिखाई देगा, एज इफ देयर इज वन ऑब्जेक्ट ओनली. सो रिजॉल्विंग पावर इनेबल्स अ इंस्ट्रूमेंट to form two separate images of two objects which are kept at a very short distance. Let us name this distance as D here, and the size of aperture let us rename it as A. The distance between these two objects is D, the size of the aperture is A, the angle formed by two diffraction pattern. इसका इमेज हमें यहाँ दिखेगा, इसका इमेज हमें यहाँ दिखेगा. If we are getting this diffraction pattern, इसका अलग इमेज, इसका अलग इमेज, it means we are able to form two separate images of two objects. It means they are finely resolved. दो अलग अलग इमेजेस बन रहे हैं मतलब रिजोल्यूशन हो रहा है. अगर दोनों मर्ज हो रहे हैं तो हमें एक ही इमेज दिखाई देगा. And sometimes you may also have the image like this and like this. They are also resolved but not completely. In that case we may get a image like this. फिर भी ये इतना वाला पार्ट हमें merging में दिखेगा. So until the image is not completely resolved, it will not be seen as distinctly. ये वाले पैटर्न में हमें एक का इमेज ऐसा दिखेगा, एक का इमेज ऐसा दिखेगा. They are perfectly resolved, right? So resolving power is the ability of any instrument to form two distinct images of the objects which are kept very close to each other. For example, if we take the case of compound microscope. Now in compound microscope, bio students are observing the slides, the cells, the animal cell, plant cell and all the bodies inside the cells are so finely apart from each other, इतना ज़्यादा close होते हैं. If there, because light is coming and light has a nature of getting bend around the corners, तो सब कुछ उन्हें merge दिखाएगा. They will not be able to resolve where is mitochondria, where is cell, where are Golgi bodies and all. So until the optical instrument is not completely resolved, we cannot get a clear image of the object, right? So we may define that resolving power is the ability of an optical instrument to resolve or to form two distinctly separate images of two objects which are kept nearby, right? Now assuming that it forms an angle delta theta, the distance between these two objects, it could be in terms of angle, it could be in terms of the linear distance. This is called as limit of resolution. दो ऑब्जेक्ट्स के बीच में जितना डिस्टेंस होता है, जिन दो ऑब्जेक्ट्स की हमें इमेज बनानी है, उस डिस्टेंस को हम बोलते हैं लिमिट ऑफ रेजोल्यूशन, राइट? एंड इट इज फाउंड एक्सपेरिमेंटली दैट रिजॉल्विंग पावर इज इनवर्सली प्रोपोर्शनल टू द लिमिट ऑफ रेजोल्यूशन. If this distance is less, resolving power is more. If this distance is more, resolving power is less. So this is what we are going to find, we are going to do in this topic, right? And this angle is found to be, yes, the value of this angle delta theta. Now this the formula, only the final expression is there in your syllabus, so I'll write down the final expression as delta theta is found to be 1.22 lambda upon a. This is the value of delta theta. This is the value of limit of resolution. 1.22 lambda. Lambda is a wavelength of light which is falling, which we are using in our experiment, which we are using in our observation. And A is the size of the aperture through which the light is bending and diffraction is taking place, right? So for all the optical instrument, whether it is telescope, whether it is compound microscope, we are going to use delta theta as 1.22 lambda by A. A is the size of aperture and lambda is the wavelength. Thank you, children.